everyone, and welcome to today's STERIS Tech Talk on the fundamentals of bioburden testing. STERIS Tech Talks are a series of webinars covering subjects relating to gas and radiation sterilization processing and the laboratory testing and validation services which support these processes. My name is Ashley Maroot, and I'm the Associate Product Manager for STERIS Applied Sterilization Technologies. I'll be the host for today's event. Our presenter today is Jason Rogers. Jason is a Principal Scientist for STERIS AST, representing STERIS on several AMI committees, including microbial methods and sterility assurance. He graduated from the University of Minnesota Twin Cities with a bachelor's degree in biology. Jason has over 15 years of experience in the medical device and contract laboratory testing industry. All attendees are on mute for the presentation. However, we would like to encourage everyone to submit questions using the questions function on the GoToWebinar control panel. Questions will be answered following the presentation. Today's presentation will be recorded and uploaded to our STERIS Applied Sterilization Technologies YouTube channel. Please note that continuing education credits are not provided as part of this webinar. And now over to Jason to begin the presentation.
Thank you, Ashley. Welcome, everyone, and thank you for joining us for today's Tech Talk on the fundamentals of bioburden testing. I'm Jason Rogers, Laboratory Principal Scientist with STERIS AST, based out of our Brooklyn Park, Minnesota facility. First, let's review some key terms that will come up throughout the webinar today. Colony-forming units are a microbiological unit of measure. That is an estimate of the number of viable cells in a given sample. In theory, during incubation, a single cell replicates on growth media to give rise to a visible colony. These colonies can then be counted to give an estimate of the viable contamination on the sample. Bioburden is the population of viable microorganisms on or in a product and/or sterile barrier system. Our bioburden estimate is the value established after applying a correction factor to the bioburden count. And our bioburden correction factor is the numerical value applied to a viable count to compensate for the incomplete removal of microorganisms from a product. Recovery efficiency is the measure of the ability of a specified technique to remove, collect, and/or culture microorganisms from a product. This is a percentage and is the inverse of the correction factor. Culturing conditions are the combination of growth media and/or incubation used to promote germination or growth of microorganisms. Note that the manner of incubation can include temperature, the duration or time, and any other conditions specified for incubation, such as aerobic or anaerobic conditions. And then finally, our sample item portion, or SIP, is a selected representative portion of a device or product that is tested. So now let's look at some general information around bioburden testing. Patient safety is the main goal of medical device manufacturers. Bioburden control and monitoring is a key component in achieving quality assurance of a manufacturing process and a consistent challenge to the sterilization process. From the bioburden test, we can determine the quantity or number of viable native microorganisms present on or in a medical device, a component of that device, or a substrate or chemical. The bioburden population can also be utilized in establishing dose for specific types of sterilization validation. The use of the bioburden data can affect the need or criticality of bioburden method validation. Bioburden is considered an estimate, as not all culture conditions can be evaluated, and not all organisms can be cultured. In addition to establishing a count of the microbes, the types of microorganisms observed on a product are critical to the quality control of the manufacturing process and maintenance of a sterilization validation. Characterization of the bioburden can inform risk assessment related to sterilization. There are varying degrees of characterization that can be performed, which will be presented later in this discussion. Monitoring and characterization of bioburden can give additional insight to the potential presence of bacterial endotoxin. Bacterial endotoxins can affect patient safety for certain device types, even after the product has been sterilized. Bioburden data can be used in a number of ways. It can be used for monitoring routine microbiological control of the manufacturing process. Bioburden testing can be implemented as part of an incoming inspection of raw materials or components or packaging that can go into the manufacture of a product. Bioburden might be used for determining comparison or equivalence of vendor materials or suppliers. Bioburden can also be used as part of the validation or revalidation of a sterilization process. Bioburden testing of component materials may also be leveraged as part of investigations for native product sterility positives or exceeded product bioburden alert and action levels. Bioburden can also be used in the assessment of the efficacy of a cleaning process or as part of an overall environmental monitoring program. Bioburden testing is important not only in determining microbiological quality and cleanliness of a finished product, it can also be used to establish the dose applied for radiation sterilization methods, such as gamma, E-beam, and X-ray. Under this process, the bioburden data is used in conjunction with the applicable table of the appropriate irradiation standard to determine a verification dose. This verification dose is then applied to the product prior to sterility testing for substantiation of a minimum routine radiation sterilization dose. Once the dose for effective radiation sterilization has been established, bioburden data will also be required for ongoing monitoring of products as part of the periodic dose audit process. An understanding of product bioburden is also an important aspect of quality control maintenance of sterilization, terminal sterilization by gaseous methods such as EO, moist heat, or VHP. Characterization of the bioburden can inform us about the comparative microbial resistance to a sterilization process for the device or material being tested. Bioburden alerts and/or action levels can point out possible changes to product manufacturing before it impacts the sterilization validation. Or bioburden can also be used to characterize the microorganisms present on a product, which can inform the manufacturer of potential presence of toxins. For example, gram-negative bacteria can lead to the presence of bacterial endotoxin on products, which can impact product release or require a product batch to be rejected. Bioburden testing is performed according to standard ISO 11737-1, which is the sterilization of healthcare products, the microbiological method determination of a population of microorganisms on products. The standard outlines processes for bioburden testing, including the testing for bioburden adverse or inhibitory substances, bioburden recovery efficiency testing, and other bioburden-related information, such as guidance on product families for bioburden testing and data use and trending. Bioburden testing should be performed routinely on a scheduled frequency, which is determined either by sterilization standards, such as ISO 11137 or 11135, or by the manufacturer themselves. The frequency should be justified to ensure that the manufacturing process remains under microbiological control, and this justification should be demonstrated from trending, risk-based assessment, and documented within the manufacturer's quality system. When looking at frequency and monitoring of bioburden, seasonal variations should also be considered. In a medical device manufacturing environment, the main contributors of bioburden contamination are from the following sources: personnel, raw materials, components, assembly processes, manufacturing environment, or the lack of environmental control. For instance, the lack of positive pressure in a clean room environment, or the lack of particulate control within that environment. Cleaning processes or poor cleaning practices can affect bioburden. Manufacturing aids such as water used in a manufacturing process. To control bioburden on a product, the microbial impact from these sources should be assessed and understood. Now let's consider the bioburden test samples themselves. Firstly, the sample should be representative of the manufacturing lot. If the manufacturing lot is specifically made for testing, then extra care must be taken to ensure the batch is representative of routine monitoring. That is, samples are representative of the normal or routine manufacturing process. For critical bioburden measurements, such as manufacturing quality or sterilization related, testing should be carried out on product that has undergone the full packaging process. Product can be grouped into the following families based on the following: device size and design, raw materials, manufacturing processes, and the manufacturing location, environment, or people. For each product family or master product can be selected. A master product can be selected. The product chosen should be representative of the whole product family. If products in the family are considered equivalent, these representative products can be chosen for routine monitoring of the family, or other members of the family can be selected on a rotational basis. The rationale for the selection of products included in the family, including the choice of the master products, should be documented by the manufacturer and maintained in the quality system. For bioburden testing samples, the items within the sterile barriers may be selected to be excluded from testing by the manufacturer. Documentation of the justification for excluding these materials should be generated by the manufacturer, maintained in the quality system, and available for auditing. Such items might include informational for use packets, packaging such as zip ties or paper ties, inner pouches, labels, or carrier hoops. It is important to communicate with the test laboratory regarding portions of the device that are to be considered for testing and those that are not. A sample item portion, or SIP, is a selected representative portion of a device or product that is tested. For bioburden testing, wherever possible, the entire product should be tested for bioburden. However, if it is not possible due to the product's size, a portion of the product can be tested for bioburden. If the product SIP is used, that is only part of the sample is to be used for testing. As large of a portion as possible should be used of the product that gives an adequate representation of the entire product bioburden. When selecting the sample item portions to be taken, the product must be considered. Where the bioburden is evenly distributed, any area of the product can be taken. Where the bioburden is not evenly distributed, either the area of the product that contains the most severe microbial challenge sterilization process can be taken, or portions of the product that proportionally represent all materials can be taken. ISO 11737, Table A1, gives examples of the basis for SIP on varying product types. The product SIP must be accounted for in the final bioburden determination calculations. So now let's look at the bioburden test method validations. Bioburden test method validation is an important component of the routine bioburden testing process, as it ensures that the method to be used will give accurate data and results, meaning that we are not grossly underestimating the bioburden. Note that bioburden test method validation process may require sterile devices so that the product's natural bioburden does not contaminate the test, as a known level of artificial spiked organism may be employed. For bioburden, there are two components of the test method validation which should be considered for completion: the recovery efficiency test method validation and adverse screening or adverse or inhibiting substances screening. There are two parts of the bioburden test method validation. The first, bioburden recovery efficiency, which is the test validating the removal and culturing of organisms from the product. The test method validation for recovery efficiency is important to ensuring accurate measurement of the contamination on the device. In addition to bioburden recovery efficiency, the need for adverse or inhibitory substance screening should also be considered. This test validates the neutralization of substances that may be inhibitory or lethal to microbes during the extraction process. It is important to ensure that the measurement of contamination on a device is not impeded or impacted by substances present in the test. The completion of an adverse substance test may not always be applicable, but such a consideration should be documented by the manufacturer and maintained in the quality system. Once established, the maintenance of the test method validation for a device must be regularly evaluated for the need to revalidate. In this regular review of method validations, manufacturers should assess whether changes in the material or construction of the device could affect the validity of the test method. Changes in suppliers or vendors, changes in device design, changes in how the device is cleaned, including changes in cleaning solutions, or changes to the manufacturing process, equipment, and environment. All of these should be assessed with regards to impact to the existing method validation. Even in the absence of significant changes, a scheduled or periodic review of the need to revalidate should be part of the quality system. This review is to ensure the accumulation of minor changes has not occurred over time. So let's look more in depth at the recovery efficiency test method validation process. Bioburden recovery efficiency test is important for ensuring an accurate measurement of contamination on a device, as the bioburden extraction process rarely removes 100% of microorganisms present on a device. There's then a need to calculate an efficiency of the method that we're using and correct for that efficiency. A bioburden method recovery validation is used to determine a correction factor, which will be applied to the routine raw data to adjust the values or compensate for the incomplete removal of microorganisms from the product in a single extraction. For example, take the setting of the verification dose during the irradiation validation process. The measured bioburden contamination values ultimately determine an appropriate dose for the verification dose experiment to ensure the product will obtain a 10⁻⁶ sterility assurance level in the routine sterilization dose. Hence, the importance that data from the test method is accurate. Products can be grouped into families for the purpose of bioburden recovery efficiency method validation based on the following: raw materials, design, product size, manufacturing processes, and the manufacturing location, environment, and personnel. Again, rationale for the product grouping should be documented, and the results from the validation can then be applied to all members of the family group for future testing. Due to the variation in products that are tested, particularly device design and materials involved in the manufacturing process, it is not practical to specify a single extraction method for bioburden testing, i.e., the removal technique and culturing conditions. ISO 11737-1 Annex A provides a decision tree for selecting an appropriate method of extraction or removal of microorganisms based on the device materials or design. Here is the flow chart, which is present in ISO 11737-1 Annex A1. There are two options for bioburden test method validation identified in the ISO 11737-1 standard. The first is the inoculated method, often referred to as direct inoculation method, which uses an artificial bioburden spiked onto the product. Efficiency is measured by sample recovery against that of the inoculum spike population. The second is a native repetitive method, often referred to as an exhaustive recovery method, which uses the product's natural bioburden. Efficiency is measured by the first extraction recovery against the total recovered from all repetitive extractions. A quick overview of the bioburden inoculation method. It has a known starting population, therefore has a consistent point of reference to use for the calculation of the correction factor. It is typically used for products with low levels of natural bioburden. And as it requires an artificial means of contamination, it may not be entirely representative of the product's natural contamination. It tests the recovery of only one species of bacteria, which is the spike organism. It can be used for products that are not suitable for repetitive washing, such as gels or powders. To understand the inoculated bioburden recovery method better, let's look at a working example. The first step in the process is to inoculate the product with an artificial spike of a known inoculum level, approximately 100 CFU of bacillus spores, and allow that spike to dry. Next, the product is immersed in the extraction fluid, and an extraction process is completed. In this example, sonication followed by shaking was used. The extraction fluid is then filtered or pour plated to determine the number of CFU present in the fluid, or the number of organisms recovered or removed from the product. The CFU value is compared to the initial inoculum population to calculate the correction factor. In this example, the inoculum population was 125 CFU, and the CFU recovered from one rinse of the product was 105. The single rinse recovery efficiency was calculated by dividing 105 by 125, which gives us 84% efficiency. And the calculated correction factor is 1 divided by 0.84, which is a correction factor of 1.2. Therefore, if we have a CFU count on a filter in the routine bioburden test of 80 CFU, then we would apply that correction factor of 1.2 to the 80 CFU for a calculated bioburden estimate of 96 CFU. Now let's have a quick overview of the bioburden repetitive method. It does not have a known starting population and involves multiple rinses of the product. It is typically used for products with consistent high levels of natural bioburden. If the product has low natural bioburden levels, or there are variances in bioburden levels, the results may be invalid for determining a correction factor. As it uses the product's own natural contamination, it tests the recovery of the species of bacteria naturally occurring in the product. Due to the repetitive rinsing process, this method is not suitable for product types that involve gels or powders. So again, we can look at a working example for this method. Here, the product's own natural bioburden contamination is used. There are no artificial spikes required. The first step is to immerse the product in the extraction fluid and complete the extraction process. Again, in this example, sonication followed by shaking was used. The extraction fluid is then assayed by filtration or pour plating to determine the number of CFU present in the fluid. This is referred to as the first rinse. The product is then re-immersed in new extraction fluid, and the extraction and assaying process are completed again. This rinsing and testing process is completed a number of times. In this case, there were 4 rinses in total completed. The CFU value from the first rinse is compared to the cumulative value obtained from all rinses to calculate the correction factor. In this example, the first rinse value was 130 CFU, the second was 35, third 10, and the fourth 1 CFU, giving a total for all rinses of 176 CFU. The first rinse recovery efficiency was calculated by dividing 130 by 176 for 74% efficiency. The correction factor was calculated similarly and was determined to be 1.4. And again, if we had 80 CFU on a routine test, we would multiply that 1.4 times that raw count to give a bioburden estimate of 112 CFU. The second component of the bioburden method validation for consideration is the adverse or inhibitory substance screening. Adverse or inhibitory substance screening is important to ensuring that the measurement of contamination on a device is not impeded or impacted by substances present in the test that could adversely affect the detection of bioburden, thus giving the appearance of low bioburden test results. The substances present do not inactivate or kill microorganisms, they merely inhibit them from replicating. Based on a detailed understanding of the components in the manufacturing process, the manufacturer can choose not to perform adverse substance screening. However, a written rationale for not performing the test should be completed by the manufacturer and maintained in the quality system. So let's talk about where inhibitory substances may come from. These may be substances present on or in the product to impart antimicrobial properties as part of the device design. Examples of this would be antibiotics, coatings, wound dressings impregnated with silver or other metals. In these cases, the presence of the substances are known by the device design team and the manufacturer prior to sending the samples to the test lab, and test method development work has begun. Upfront notification of information on known adverse substances will allow the test lab to carry out trial work and build neutralization into the proposed test methods in advance of beginning the method validations. Sharing this information with the test lab along with safety data sheets will ensure that the appropriate safety and disposal processes are considered as part of initial product and method review. There may be adverse substances present on or in a product that are unintentionally cause inhibition in the test method. Examples of this may be product coatings or residual detergents from cleaning processes. Typically, the presence of these substances or their impact is unknown by the manufacturer or the test lab prior to test method development work, and is only discovered during the first phase of testing. They are often more difficult to overcome as the cause of the inhibition may not be obvious and trial work may be required to develop a solution. These unknown adverse substances are more likely to cause delays to project timelines as it is an unexpected issue. Note in general, the presence of adverse substances can cause additional testing costs, additional products may be required, and possible time delays. For these reasons, it is recommended to perform feasibility testing as early as possible in the project, as multiple attempts may need to be obtained, may be needed to obtain a successful outcome. Bioburden adverse screening verifies the bioburden test method suitability. This involves neutralization for a quantitative test. This test requires sterile products and uses low levels of specified test organisms with populations between 10 to 100 CFU to inoculate the test vessels. Adverse screening for bioburden test method suitability ensures that the product or or substances present in the product do not impact microorganisms in the test extraction or growth media, and thus creating a falsely low measurement of contamination on the device. Under the Bioburden adverse screening, products should be subjected to the same extraction technique planned for routine testing and validated during the Bioburden recovery efficiency method testing. The required outcome in the Bioburden adverse screening test is that the quantity of growth or number of CFUs recovered from the vessels containing the product is not significantly different to that seen from the vessels containing no product, which are positive controls. There are several techniques which can be employed to overcome adverse or inhibiting substance issues in the Bioburden test, such as filtration, dilution, or inactivation or removal of the inhibitory substance by the addition of a neutralizing substance. Now let's discuss the enumeration and characterization of Bioburden. The determination of the number and types of microorganisms present on a product are the most critical components in the control of a manufacturing process and are of particular importance in the radiation validation process. For radiation, the Bioburden data on the number of microbes can be used in conjunction with the applicable table and the standard to determine the verification dose to be applied to the product prior to sterility test or the substantiation of a minimum routine dose. The types of microbes present can affect the potential resistance to radiation, that is the potential of growth in the dose audit. Understanding the number and types of microbes are also important factors in grouping products into product families, the inclusion of a product into an existing family, or the selection of a product to represent a product family. Bioburden is a quantitative test. As a result, we will have a specific number of organisms which are reported as CFU or colony-forming units. Seen here in the picture, a number of different colony types. Each of these growths is considered a colony-forming unit. Bioburden testing is primarily performed by cutting up, disassembling, or flushing the fluid path of a device. When cutting up or disassembling the device, extensive manipulation may be necessary to allow for placement of the sample into a sterile test container. Note that the more manipulation, the greater the possibility of contamination during testing. Sterile tools are used to prepare the sample. The test sample is placed in the test container. A measured volume of sterile rinsate solution is then added to the test container. Generally, suspension fluids such as buffered water or buffered water with a surfactant are used. To remove microbes from the device, the sample may then be extracted in a variety of methods or combination of methods based on the material rigidity and design. The extraction methods to be utilized could include, but are not limited to, sonication, mechanical shaking or hand shaking, vortexing, or stomaching. After the extraction process, the extraction fluid may be divided for culturing of different organism types. The extraction fluid will generally be assayed by either filtration or pore plating. For the filtration method, the extraction fluid is poured into a filter cup with a membrane filter in place. They are located on a filtration manifold. A vacuum pump is used to pull a vacuum. The test fluid is suctioned through the filter membrane, trapping the bacteria and fungi present in the liquid onto the filter membrane. The filter membrane is then placed on a growth-promoting auger for incubation. Pore plating is a is used where filtration method is not an option. This could be due to product debris, product particulate, gels, or items that may clog or degrade the membrane filter. Pour plating is carried out as follows. A portion of the extraction fluid is pipetted directly into a Petri plate. Then tempered growth-promoting agar is poured over the plated extract fluid. These are mixed, and the agar is allowed to cool for solidification before it is placed into incubation. After incubation, the visible colonies, or CFU, on the filter membrane or in the pour plate agar are counted. The raw CFU count is then multiplied by all applicable factors, such as the correction factor from the test method validation, any dilution factors, and accounting for the sample item portion. If it is reasonable and appropriate to do so, the bioburden detection limit can be improved by reducing the number of parameters tested. For instance, aerobes, anaerobes, sporoforms, and fungi could reduce the number of tests that we're performing there. Dual incubation of plates. We could transfer plates after aerobic incubation into a fungal incubation to reduce the amount of splitting of the sample. Additionally, pooling samples could be performed, but note that the pooling of samples for extraction can affect the ability to see sample-to-sample variation within a group of products. When looking at results, it's worth noting that difficulties may occur in the counting process, and these will be recorded as follows. Results may include indication of TNTC, or too numerous to count. So individual colonies that are indistinguishable due to the high numbers of colonies on the filter membrane or in the pour plate agar. These are values outside of a countable range, typically greater than 300 CFU. Results may also describe the presence of a spreader, which is a colony that grows over a considerable portion of the filter membrane or pour plate area and impacts the accuracy of counts. Spreaders can be caused by the nature of the organism, particulates from the product, or by fluid on the filter membrane or agar plate. Note that inverting the plate during incubation can sometimes help with this issue if it is fluid-related spreading. As mentioned on the previous slides for bioburden testing, we use growth-promoting agar and incubate in order to determine the CFU present on a product. However, several factors should be considered when choosing the culturing media and incubation conditions. The raw materials used to build the device, for example, the products manufactured from tissue or other natural materials, can be at risk of contamination with obligate anaerobes. The method of manufacturing and conditions under which the product is manufactured, the potential presence of fastidious organisms, and seasonal variation of potential contamination sources. Example would be if a product contains a natural material such as honey, which sees contamination vary across the seasons. As there's no single combination of media and incubation conditions that can support growth of all microorganisms, a general purpose non-selective culture media and incubation conditions can be used unless fastidious microorganisms are suspected to be present on the product. A rationale for consideration of the organism being tested should be documented within the manufacturer's quality system. Consideration should be given to the conditions selected to minimize the potential to overestimate the average bioburden due to counting of the same organism on different media. Table A2 from ISO 11737-1 standard specifies agar types, incubation times, and incubation temperatures. TSA at 30 to 35 C, 3 to 7 days, being typically used for culturing of non-fastidious bacteria, and SDA or PDA at 20 to 25 C for 5 to 7 days being used for the culturing of yeast and molds. Note, extended incubation can be requested if needed, if slow-growing organisms are concerned. Why would we carry out bioburden characterization? Characterization of CFEs present on the device form an important piece of the information generated from bioburden testing, and particularly for radiation, sterilization, validation process. The identification of the organisms can assist in understanding the potential challenge to sterilization dose, in grouping of products into families, or with the selection of a master product. Microbial characterization is also useful during investigation on exceeded bioburden limits or sterility failures. The most basic form of identification is colony morphology. It gives us visual description of the colony's growth characteristics such as color, shape, consistency, and edge. For example, the organism in the bottom left picture is green, wrinkled, dry, irregular, with an irregular edge. The visual description helps to classify observed phenotypes over time. When coupled with further identification processes, this can give insight into the possible genus or species recovered. Gram staining of the colony gives us further clues to the organism identification. The gram stain tells us about the cell wall of the bacteria recovered. When viewed under a microscope, gram-positive microorganisms appear purple from the primary stain in the staining process due to a thick peptidoglycan layer. Gram-negative organisms appear red from a secondary stain in the staining process due to a thin layer of peptidoglycan that is easily discolored after the primary stain. Due to the staining, the shape and grouping of cells can also be seen, and this is to assist in the identification of the organism, for example, gram-positive cocci versus gram-positive rods. To obtain a more specific higher confidence level match, MALDI-TOF, or matrix-assisted laser desorption ionization time of flight, testing can be completed and is based on ribosomal proteins analyzed by mass spectrometry. For the MALDI-TOF testing process, a smear of the organism to be identified is placed on a target plate, which is the blue plate at the top of the photo, or top of the slide. This smear is then overlaid with test matrix solution, and when dry, the target plate is placed into the MALDI-TOF instrument. The plate is electrified and subsequently blasted with a laser to separate the proteins which travel up a vacuum and are measured by a detector. Each microorganism's measured protein type and quantity ratio provide a spectra or fingerprint shown at the bottom of the slide there. This spectra is compared against a library of other known organism spectra to achieve an identification. The data from bioburden testing should be trended, alert and action levels established, and this data reviewed at regular intervals by the manufacturer for action or mitigation. When developing a bioburden sampling plan, testing frequency should be evaluated for seasonal variation. Bioburden characterization should be monitored and trended for investigational root cause analysis and assessing the risk of changes in population of resistant organisms. Manufacturers are responsible for setting their own internal bioburden alerts or action levels. They are not standalone acceptance criteria, but rather should be used as a means of monitoring the manufacturing process. The method of sterilization and the amount of overkill in the process should be considered when setting alert and action levels. The levels are typically established based on historical data and trends. When setting levels for a new product, temporary levels can be set until there is enough data to gather to establish long-term levels. Typically, a baseline is created with initial testing being performed at a higher frequency, weekly or monthly, or with each build until data is built up to create longer-term levels, and the frequency can then be scaled back. Reestablishing long-term levels once sufficient data has been gathered. Review levels at a defined frequency to ensure that they have continued appropriateness. Alert levels indicate when a process may have drifted from normal operating conditions. Typically, no actions are required, but an investigation may be initiated and a corrective action may be carried out. However, repetitive excursions may be treated like an action level excursion. Example of an alert level excursion action, cleaning of a manufacturing environment might be initiated, or increased environmental monitoring for a period of time if a bioburden level excursion is observed. Action levels indicates when a process has drifted from normal operating conditions. An investigation must be initiated and corrective actions carried out. Examples of an action level excursion. Action would be to perform a documented investigation into the root cause of the excursion with actions to mitigate the findings of that investigation. Common methods used for setting alert and action levels. Trending of bioburden averages or means, coupled with standard deviations, maybe 1 to 2 standard deviations for a alert level and 2 to 3 standard deviations for an action level. Additionally, a Short chart might be used. There's additional information on that in ISO 7870-2, or cumulative sum charts, which is in 7870-4. When setting alert and action levels, consideration should be given to detection levels as well as TNTC or spreaders.
Thanks again for joining us today. This concludes the webinar.